Hi, everyone. Thanks for being here. I'm excited to talk today a little bit about my work, bridging computational approaches and biodiversity science. And I'm going to start by giving a brief motivation for this work. As you may know, we are in the middle of what is being called a biodiversity crisis, or the 6th mass extinction. We are losing species at an alarming rate. And extinction rates are orders of magnitude higher than the background rates of the past. And this is happening for a variety of reasons, which I've tried to summarize here. So climate change, habitat loss, invasive species, and overexploitation of species that are considered resources. And I'll just note that this isn't just a big deal to scientists or environmentalists. The World Economic Forum actually listed biodiversity loss as the 4th greatest threat to our world economy. So that was behind infectious disease, climate change, and weapons of mass destruction. So understanding what's happening to our biodiversity and what we can do about it really has meaningful impacts on us as people living on the planet. So understanding these drivers and their impacts is a pretty monumental task, not just because they are so pervasive and interacting, but because the systems they impact are themselves inherently complex. So this is sort of the Ecology 101 diagram of ecological complexity, where we have individuals making up populations and populations interacting to form communities, which depend on the abiotic factors which make up ecosystems, which are all part of biomes. So these drivers of biodiversity loss interact with each of these pieces differently, with cascading effects up and down the levels of complexity. And as you can imagine, it is quite a task to decide how to quantify all these levels of complexity in a way that's meaningful but still tractable. So for a variety of reasons, both I think conceptual and logistical, this huge scope of biodiversity is often boiled down to just a few species-based metrics. So whether that be species richness, which is the count of the number of species, or species diversity, which incorporates the number of individuals of each species, we almost always summarize biodiversity by these or similar metrics. And there's been a lot of recent work thinking about whether or not these species-based metrics are actually capturing the kinds of changes that are happening as a result of these drivers of biodiversity loss. So this is a figure from a quite high-profile paper in 2014 that took a whole bunch of different time series of communities. So these are counts of species and individuals through time to get a general picture of local scale biodiversity. Each one of these lines is a different community, and there's a lot going on here, but the main picture is in the 2 black lines, which give the overall mean trend line for these 2 different metrics. And in the left panel, we have a plot of the log of species richness, so the count of species. And you can see that this overall trend line is not significantly different from 0. So despite the fact that we have some really precipitous gains and losses in different communities, the overall trend was that there is no change in local species richness. You can see, though, in the right panel, which is measuring Jacquard similarity, so how similar is the community to what it used to look like, that there is a significantly decreasing similarity through time. So even though communities as a whole are not generally gaining or losing species, the composition of communities is changing, the identities of those species. So how can we better understand what that composition means? And my work has really centered on functional diversity as an alternate lens for understanding biodiversity change. Functional diversity is potentially really powerful because it doesn't just count the number of species, assuming that they are all equally important, but it instead summarizes the characteristics of those species that are determining the ecological role they play. So this is my favorite little toy example. These are fig wasps on a fig fruit. Fig wasps play a really unique role in that they're one of the only insects that can efficiently pollinate fig flowers. So in a species-based approach, losing or gaining a fig wasp would be just the same as losing or gaining any other species. But from a functional lens, the loss of a fig wasp would be a huge reduction in diversity because they have a set of unique traits that allow them to perform this unique function. So I've been curious to answer this general question, how do functional approaches detect change in communities beyond the species-based metrics? And I should say that functional diversity approaches have been a really exciting idea in ecology for a while, but looking at functional diversity on a large scale across a lot of communities hasn't really been possible due to both data and computational limitations. So I'm going to talk about my work in both of the data and the computational avenues. So from the data perspective, there's been a lot of work recently trying to compile data on functional traits. Functional trait data is pretty costly to collect because, as you can imagine, it requires measuring specific traits on individuals. And so there have been efforts for different taxonomic groups to create species level trait databases. And then there are also a few different compilations of time series, so looking at assemblages through time. And my goal was to try to bridge these two data sources to create a time series of functional change. So this is conceptually simple, but in practice, not so easy. And we have this pretty involved data processing pipeline. And I'm going to talk about just one of the issues that we had to address, which is dealing with taxonomic inconsistencies. So whenever you're working with biodiversity data, one of the challenges, especially when you are bringing together multiple data sources, is that the taxonomy, or basically what species you decide an individual is constantly changing. So whether that be one species splitting into two species, or two species merging, or one species being called something by one group of people and being called something else by another group of people. You could have a sample in one data set that is from the same species as another data set, but is being called something completely different. And there are taxonomic databases built to sort of resolve these inconsistencies, but there hasn't been a way to access them in the past that allows you to resolve large volumes of names. So my advisor and I developed an R package to do just that, to access existing taxonomic providers and provide an interface for accessing that data. And this is a little example of how that might work. So we have two different data sources, IUCN classifications on the left and body mass on the right, and a whole set of species. So the sort of intuitive way you might approach this is to just join on the species names. And when we do that, we see, oh, darn, there's only one species that occurs in both of these databases. But then when you resolve each of those species names to the common ID from a taxonomic provider and then join on that ID, which is what we did over here, you can see that, oh, actually, there were some similar species just being called something different. So, and we do have data for quite a few species. So as you can imagine, these discrepancies can really be magnified in these big data sets. So it's important to keep track of them for larger data sets. And then there were a whole bunch of other things we had to deal with in terms of standardizing sampling and sampling effort through time. But the long and the short of it is that in the end, we had almost 2,500 individual time series from 53 different studies for amphibians, birds, and mammals. So I don't know if this looks like a lot of data or a little bit of data to you, but the sort of next best thing we had previous to this was just a few time series for a single taxa. So this does represent a pretty significant contribution to the sort of library of functional time series that we have. And then to look to quantify functional change for each year in each time series, we calculated a few different metrics of functional diversity. So the way you can think about a functional trait space is as a multidimensional space with an axis for each measured trait. So here on the left, there's a little example figure of 3 different traits: wood density, seed mass, and specific leaf area. And functional richness, the metric functional richness, simply measures the volume of that trait space. Functional evenness measures how species are distributed in that space and how individuals are distributed across species. And divergence measures the distance of each species from the center of that multidimensional trait space. So these 3 aspects measure complementary aspects of the trait space and hopefully give a pretty complete view of the functional change that is happening. So here is the first figure with my actual data. And this is reproducing the figure from that 2014 paper that I showed you at the beginning. And so the first question was, well, does our data reproduce those species-based patterns that they saw? And yes, it does. So on the left, we have species richness, just like in the previous figure, and the overall trend line in black. And the line itself is a little bit deceptive because of the scale, but it is non-significant. So there is not a significant trend in the number of species over time. And on the right, Jacquard similarity. So there is significantly decreasing similarity. So communities are looking less like themselves over time. So great. What does that mean for functional diversity, which is why we're doing this whole song and dance? So these are 2 of the functional diversity metrics that we calculated. And surprisingly, what we found is that for all 3 metrics, only 2 are pictured here, but it's true for the third. There was also no significant change through time. So despite the fact that many communities are experiencing huge increases or huge decreases in functional richness, evenness, and divergence, the overall trend is 0. So I think a few big take-home messages about this, or rather next steps, The first is that there are huge functional shifts happening in these communities, but we don't really have a good grasp of what a normal amount of functional change is and what it would mean for there to be a huge increase or a huge decrease in these functional richness. So there is a big opportunity here to go from this broad scale sort of summarization of a whole bunch of communities back down to the community level to look at specific community characteristics. And one of the potentially really powerful things we could do with that is to establish some general rules that predict different scenarios of functional loss, and particularly when functional loss or functional gains could accompany different species level shifts. Yeah, so I'm just going to finish with some acknowledgements. My advisor and committee, and of course, Krell, which has really made a huge difference in the path my PhD has taken. And they're not pictured, but my lab and department, which have been a great home over the course of my PhD. Yeah, so thanks for listening and happy to take questions now.